Thank you very much for the opportunity to give this talk. My name is Menden. I'm a junior group leader in the Institute of Computational Biology, and I would like to present to you today predicting potent drug combinations by exploiting monotherapy resistance. For those who are not working in oncology, just a short introduction to cancer evolution. We all start with a healthy genome, but during our lifetime, we accumulate various different somatic mutations, which may be induced by smoking or UV sunlight exposure and ultimately lead to cancer. These tumors are not shrinking on their own. Actually, after diagnosis, often we have suitable treatments to get them under control. However, there's always this tiny cell population, these few clones which are resistant to the initially very successful treatment. It becomes very challenging to get them under control with a follow-up treatment. And at some point, it may even lead to a metastatic disease, so meaning spreading to other parts of your body. And then cancer becomes a very difficult disease to cure. So the question is not if we get cancer, it's rather when. And there are promises, but also failures in precision medicine. And here I want to give you an example of a melanoma patient, which a picture which traveled around the world, a patient who had lesions all over his body. And this skin cancer is often driven by, is predominantly driven by the MAP kinase signaling pathway on the left-hand side, just a cartoon, EGFR, KRAS, BRAF, MEK, ERK, which ultimately lead to cancer cell growth. And 60% of these patients have a BRAF B600E mutation, which leads to very strong activation of this pathway, independent of upstream signaling. And we have for this very potent drugs like vemurafenib, which basically inhibit this BRAF V600E mutation. And you can see extremely strong drug response in this patient within 15 weeks, which happened to be also a BRAF V600E mutant patient. However, just 8 weeks later, you see very strong relapses, drug resistance. And this could happen, for example, due to an MEK amplification, that you get, again, this pathway activated, reinitiated through an additional mutational event. So the aims of my ERC is to understand these resistance mechanisms, identify them, and come up with smart drug combinations to overcome them. And in clinics, that's actually showed large promises to actually do a double hit on this pathway, or even a triple hit, and also inhibit EGFR. So here an example of another cancer type. Before we talked about skin cancer, this is lung cancer, lung adenocarcinoma, and gefitinib resistance. Gefitinib is an EGFR inhibitor, which is given to lung adenocarcinoma patients, which are activated for exactly the same pathway, EGFR, KRAS, BRAF, MEK, ERK, leading to cancer growth. And this EGFR mutation is basically it's a receptor tyrosine kinase, so which becomes independent of ligand binding and always sends signal when it's mutated. And this could be activated by exon 19 deletions or a specific mutation in the activation loop, the L858R mutation. If you give gefitinib, which is a potent EGFR inhibitor, you annihilate the signaling. However, due to cancer evolution, it's such a strong evolutionary pressure, and they're so addicted to this pathway that they often obtain this TC90M mutation. And this mutation is rendering the drug entirely useless. And what is happening there? It's changing the binding pocket of the drug so that the small molecule can't bind efficiently anymore. And here in the spinning molecule, in the spinning protein that's EGFR, you can actually see how it would not bind if this red amino acid is exchanged. And you may notice on the bottom the references. This is more than for 15 years known. So why is it still interesting? It took more than 10 years to get that into clinical practice. So nowadays, gefitinib became again the first in-line treatment for metastatic lung cancer patient if they don't have a T790M mutation yet. And this is really embracing also resistance markers and not only focusing on the sensitivity markers, which are attractive because that means your drug works, you have a patient cohort you want to sell the drug to, but also to understand resistance mechanisms and say when it's not working, which can open opportunities. This year also we developed a new method to identify these resistances in drug high throughput screens systematically, published in Cell Patterns. And aims of my ERC starting grant is to first identify drug resistance, but also identify then smart drug combinations to overcome it. And that is a very challenging task. Just looking here at this signaling network, where to begin when you want to basically find the best drug combination to target this hairball of information. And here in the red circle on the right hand side, that is the publication we published in Nature Communication. I would like to talk about now a bit more about drug combinations, which is AstraZeneca Sanger Drug Combination Prediction Challenge. It's a crowdsourcing challenge, which we ruled out to the whole bioinformatics community, where we first had a design phase looking at baseline models. Is it feasible? Is it predictable? Is there a signal in the data? Split it into train and test set. Proposed a scientific question to the community in the open challenge phase, which is roughly 2 to 3 months. They try to build their models to predict the outcomes on the test set. And then we have an unbiased evaluation to see who is the best performer. And we had more than 80 methods submitted to that to actually see, to derive some rules why the best performers perform best. And this meta-analysis took more than another year to actually conduct, to really tease apart all this information. We have been involved in a couple of challenges also on the organization side, but also participated in drug sensitivity, drug combination, toxicity, and gene essentiality. The AstraZeneca drug challenge, which was led by us, that is, we looked at 85 different cancer cell lines from 6 different cancer types. And on the left-hand side, you see how the pharmacology data is normally looking like. You have this 5x5 drug response matrix. On the first row and the first column, you have the monotherapies. From the monotherapies, you can derive a theoretical surface, here with a Löwe additivity model, but could have been also Bliss independence or another model. And then you compare these two different surfaces, the experimental surface and theoretical surface. And depending on if the volume is positive or negative, you call it synergistic or antagonistic. If they would be entirely identical, then you would call it additive. And we had data for more than 100 unique compounds, more than 900 combinations, and more than 11,000 experiments. And in addition to this, we provided the participants in the challenge with a deep molecular characterization of these 85 cancer cell lines, including mutational information, copy number, DNA methylation, and gene expression. And the challenge was predict synergy. So maybe the most interesting question, how did the best performer actually do it? And she used data from GTSC and CCLE, which are the 2 largest cancer cell line panels currently available with deep molecular characterization. She used gene expression, copy number, mutation, methylation data, and augmented all these features depending on a protein-protein interaction network. and she anchored on the drug targets and then augmented their features accordingly. And this target-centric view was common across all the best performers. So commonly, what we did, or what was done so far, is looking in a disease-centric view when you try to predict drug response. So you have a cancer cell line, you molecular characterize them at the cell line panel, you filter maybe for human relevant mutations to get rid of cell line artifacts. Then you can filter maybe for cancer genes to not look at too much noise. And you come up with these kind of binary Venn matrices, which contains mutation information, mutant or wild type, for these genes. And then you try to predict the drug response. In the target-centric and the drug target-centric view, we want to look also upstream and downstream of drug targets, what's going on, regardless of if it is an oncogene or tumor suppressor. We want to also learn a bit more about what the drug is doing in this network, in the signaling network. Basically, another thing what was unique or common across the best performers, they used kind of ensemble methods. They used multiple methods to make a prediction to get robustness in the prediction. So to basically decrease the variance and the noise. The algorithm doesn't matter. That was a very surprising finding. So all roads lead to Rome, literally. It doesn't matter if you use a linear regression, a decision tree, AI, artificial intelligence, Gaussian processes, random forest, or SVMs. There was really no benefit of either. Also, this may come surprising to some, AI doesn't solve it. So AI is not the silver bullet for every task. And looking at the best performers, we realized all of them were looking at cancer genes. So they were filtering from these 20,000 genes, just looking at cancer-relevant genes, and filtered with that a lot of noise in the data. And they only focused on driver variants. So in mutations which have been reported to be functional in cancer. So removed all kind of passengers and artifacts in the human cancer cell lines. So how can we get biomarkers of synergy? That is basically, for me, very interesting for the simple reason you don't only want to have a model which is predictive, you want it to be also interpretable. I want to go even a step further. You also want it to be translatable to the clinics. And this is a very difficult thing to ask in a challenge because we don't have any gold standards. We don't have any synergy biomarkers which are used in clinics yet. So we thought like, well, we can't get a objective scoring function for that. Maybe we can ask the participants. So we used a survey monkey. Unfortunately, we got a lot of submissions there, which were really hard to interpret because we made it mandatory. So they just dumped a lot of data in there, which was not interpretable and by far not translatable. So in the manuscript, in the paper, in the meta-analysis, we decided to we use a different strategy. We approached the 3 best performers again and asked them, can you please give us a list of important features for your predictions? So we looked at the predictive models and their feature importance. And in a random forest, which was used by one team, that can be rather easy derived because you have this kind of decision tree and each of these decisions is a gene. Is it mutated? Yes, no. Then you go either left or right. And as closer you're to the root, as more important your feature is. And since you have a lot of these trees, you can just average them and you can get a ranked list of your features. Other methods, other teams used more complex methods like deep learning, where it's a bit more harder to extract the features. And they, for example, employed a method that they left out features and then to see how does that impact the performance of the model, which is great, but comes with a caveat: if you have co-correlated features, you might not accurately rank them because another feature could compensate for the loss of another one if they're co-correlated. However, we basically looked upstream and downstream of these of the drug targets if there were features related to the drugs. So if we have a biologic interpretation for these features, if they had a GO-term similarity to the drug target, or if for any unknown reason some of the top performing teams consistently picked that feature and we don't know why, then we also wanted to investigate it. And this hypothesis we tested with basically an extensive literature review with additional experiments in AstraZeneca and also with publicly available drug combination data from Merck. And that led to 13 high confidence synergy biomarkers. Here's an example of a BP2 amplification, which is a synergy biomarker of EGFR and AKT inhibition. So when ABP2 is amplified, which is also known in clinics as HER2, it's strongly activating PI3 kinase AKT signaling. And you can block downstream AKT, which is a quite reasonable treatment choice. However, if you also inhibit EGFR, you prevent this cross-activation. And this was predicted to be synergistic, particularly in the amplification of ABP2 scenario. And indeed, we observed that in our data. Here you see on the y-axis the synergy scores. As higher it is, as more synergistic, as better these two drugs are working together. And if you have the mutant ABP2, in this case, it's an amplification, they are much more synergistic than the wild type. We also observed exactly the same thing in completely independently tested cell lines in a Merck data set. So it was experimentally actually validated significantly in another data set. So these results were really promising that we really want to follow this route on how to come from resistance to drug combinations and how to overcome them. But I also would like to use this opportunity to generalize the core mission of our lab, of my lab, which is to explore the deep molecular characterization of all kinds of biological models, not only human cancer cell lines. We also look at PDX models, genetically engineered mouse models, and even patient data. So we also look at clinical trials. Then with deep molecular characterization, we marry with phenotypic data. In most cases, this is a drug treatment, a drug response. But this could be also, for example, a CRISPR screen where we look at drug targets or any other phenotypic readout. And then we try to identify biomarkers for this. And this is always in the theme of precision medicine, that we can stratify patient cohorts based on biomarkers for treatments or for risks of developing a disease. And for this, we use lots of machine learning, but also biostatistical methods. And what I mean by generalized, we have applied that to many other disease segments, including diabetes, COVID-19, Parkinson's disease, inflammatory skin diseases, ferroptosis, and also more upcoming also in tuberculosis drug resistance, where we obtained one other grant and another one is currently under review. With this said, I would like to acknowledge my ERC collaborators. The whole ICB, this is just a short list of a few PIs, Fabian Theis, the director of the Institute, Carsten Marr, Maria Colomb, Matthias Heinig, and many more. So I can't list them all, but it's an amazing environment to be in. HMGU, the Helmholtz Center in general, with Elis Egini, who gave a lot of feedback also for my ERC application, the Menden lab, which actually enabled all this, and SBR and ICB office also for the administrative support. With that, thank you very much for your attention, and it was a pleasure to be here today. Thank you.